myself. Okay, so we're going to start today's discussion. Make sure you sign in for roll call. I'll make sure that keeps going around. And then I'm going to share screen. Okay, so I think we're good. So I already uploaded the annotated notes for you. You guys can see I went ahead and drew all over the place. These are already on CAT courses in that files, TA resources for students, and then Maria's annotated discussion worksheets. You have all this. I drew stuff out. I wrote explanations. I gave you a bunch of rules. Secondary structures, you have alpha helixes and beta sheets. You have to memorize those rules. What are the rules for you to make an alpha helix versus what are the rules for you to make a beta sheet? You need to memorize those rules. So as I was reading this chapter, they like sprinkled the rules throughout the entire chapter, which was really annoying. They didn't have like a table at the end summarizing all the rules. That would have been beautiful. They didn't do that for us. So I tried to get everything that I could. And I went ahead and organized it for you right here. And that is why we have just alpha helix and then all the notes that I found for alpha helixes and then beta sheets, all the notes that I found. There was a little bit less. So make some flashcards, whatever you have to do. But what I'm going to do right now is just cover them really briefly. For an alpha helix, you have about 3.6. Let me draw a little alpha helix here. You have about 3.6 amino acids per turn. 1/4 of all amino acids that are found in alpha helixes are alanine. A chain of glutamic acid is negatively charged. So if you guys remember D and E, those guys are negatively charged. If you have a chain of those, the charges will repel each other and that will prevent an alpha helix formation. So if they give you like a bunch of Ds, you know those are negatively charged and they ask you, is this going to form an alpha helix? Yes or no? You're going to say no. And if they ask you why, you're going to say because the charges repel each other. You can't make a stable alpha helix, okay? Oh, let's see. Same for positive charges. If they give you, what did we say was positively charged? KHR. KHR. If they give you a chain of R's and they're positively charged and they ask you, is that going to form an alpha helix? You're going to say no, because the positive charges repel each other and you cannot make a stable alpha helix. Glycine is more conformationally flexible. Why? Because glycine is the tiniest amino acid. It only has a little hydrogen for its R group. So that is beautiful. Alpha helixes, they love glycine. No, wait, glycine more conformationally flexible and proline. Wait, maybe they don't like glycine. I don't remember what I wrote. Is it glycine and proline? Okay, I gotta straighten that out. So just like remind me, let me put a little star right here because I have to figure that out. But proline for sure. Alpha helixes do not like proline. Let me put a little star right, or let me just underline it here. Just remind me before we go to like straighten that out. I gotta look it up in the book because I don't remember what I wrote. But basically, alpha helixes do not like proline. If you ever see like 3 prolines in a change and they ask you, is that an alpha helix or a beta sheet? You're going to say beta sheet because beta sheets actually love proline. Okay. So you have to know that. Why in an alpha helix? Because proline is rigid. It doesn't allow rotation. It's destabilizing. Thank you. Everyone signed in? Yeah. Okay. It's destabilizing. So it tends to inhibit. So that is why alpha helixes do not like proline. And then one common thing that you find in alpha helixes is that you'll have a positive charge and then 3 residues down, 3 or 4, you'll find a negative charge. That's what you often find. It's like a kind of like a trend that you find. Because they will create an ion pair. So that's why for the alpha helix, okay? So let's see. You usually find negative charges at the amino terminus. So we know that for a chain of amino acids, you have like the amino end, which is the beginning, and then you have the carboxy end, which is the end, right? So at the beginning, the amino terminus, you usually find negative charges. And then towards the end, you'll find a positive charge or positive charges, okay? So remember, they're just gonna give you, like if they give you this on the exam, they're just gonna give you like lines of letters, and you have to determine, is this an alpha helix or is it a beta sheet? That's why you have to have your rules memorized, okay? Let's see. Beta sheets. The backbone zigzags. So you have two different types of beta sheets. You have type 1 and type 2. In type 2, you normally see glycine. And then in type 1, you normally see proline. And proline is 2 times more common in turns. And you have 2 types of beta sheets. They're parallel, and then you have anti-parallel. And the anti-parallel are twice as common as parallel. Okay, so let me just quickly draw those for you. So for beta sheets, you have the parallel, which is they're all going in one direction. And then the anti-parallel goes like this, opposite directions. And then for, see, okay, so they have alpha helix, beta sheets. That's what the beta sheets look like. All right. Let me look for alpha helix rules really quick. Alpha helix rules on glycine. Just want to make sure we know what's happening. So here's the NIH. This is a good reference. A predictive rule for protein folding is presented that involves 2 recurrent glycine-based motifs that cap the carboxyl termini of alpha. So it seems like they do.
Really?
So a predictive rule for protein folding is presented that involves 2 recurrent glycine-based motifs that cap the carboxyl termini of alpha helices. So it's saying that glycine is at the end, basically at the carboxy. In proteins, helices that terminated in glycine residues were found predominantly in one of 2 motifs. These glycine structures had a characteristic pattern of polar and apolar residues. Visual inspection of known helical sequences was sufficient to distinguish the 2 motifs from each other. These glycine motifs in which local sequence available. Okay, so it looks like it's good. Stop share. Okay, I'm gonna stop sharing for a second. I'm gonna put you guys on pause. Unless record first, then share screen. Okay, all right, so draw the polypeptide, KSFAD. Well, I went ahead and drew it right here for you. Now, anytime you have to draw a polypeptide, you have to follow the rules NCC. So I'm gonna draw on the board. So anytime you have to draw a polypeptide, you follow the rules, NCC, NCC. So this is 1 amino acid, and then it holds hands with the next amino acid, and then it holds hands with the next amino acid. Okay, the first N is the nitrogen for the amino group. The first carbon is the alpha carbon for that amino acid. So the R group has to come out of here. The second carbon is the carbonyl. And is that easy? That's the backbone. So we have our amino, alpha carbon, carbonyl, amino, alpha carbon, carbonyl. So let's go ahead and draw that out. So I'm going to go ahead and use black for my backbone. So we're going to start here with our N, and then we're going to go C, C, N, C, C. So then down here, we're gonna do the numbers. So this NCC, this is 1 amino acid. This NCC, this is 2 amino acids. And they asked us to draw 6 amino acids. So we just keep it going. N, C, C, N, C, C, N, C, C. How many do I have? So this is 1, 2. So from this N, these 2 C, this 3, 4, 5. I've got 1 more. Is that easy? Yeah, you can do that? Okay, cool. Now, once you've drawn your backbone, oh, okay, we have to finish the backbone. So let's go ahead and I'm gonna use orange now. So NCC. So our first C is the alpha carbon. The other guy is the carbonyl. And then every nitrogen is going to have a hydrogen. So remember, a nitrogen can be attached to 3 things, 1, 2 bonds here, and it's missing 1, so you just add a hydrogen. So the first carbon is alpha, the second carbon is carbonyl, and then we have our nitrogen. We have our nitrogen, we just add a hydrogen. Okay, so then the first carbon is alpha, the second one is carbonyl, the nitrogen, we just add a hydrogen. Okay, then we have our alpha carbon, carbonyl. Is this getting easy? Yeah? Okay. So then the first carbon is alpha, carbonyl, hydrogen. Alpha, carbonyl. Oh, okay, so this is the last one. So it's not a carbonyl, it's COO minus. It's supposed to be COOH, but at physiological pH in our bodies, that's about 7. And we know that at a pH of 7, this guy already donated his proton, so that's why he's minus. And so now we're going to kind of start labeling. And so this is the amino termini. You have to know the names of things. So this is the amino termini. This is the carboxy termini. All right. So now, if they ask you what thing here is a proton donor and which one is a proton acceptor, well, this guy has this nitrogen has a hydrogen, so that is a proton donor. And then this guy right here is a proton acceptor. We have to remember that oxygen has a slight negative charge, and so it's going to go ahead and accept a positive charge that comes along with a proton. Okay, so and you also have to remember that this is a chain of amino acids. When it becomes a protein, it's going to fold into its 3-dimensional structures, and then it's going to interact with other things in the cell and with itself while it's folding. So that's why it's important to know where these interactions are going to happen. So every oxygen from the carbonyl is going to be a proton acceptor, and every hydrogen from the nitrogen is going to be a proton donor. Okay, so, okay. Next, all right, we got to learn some rules. We got some angles. So we have phi and psi angles. So this is phi and this is psi. Now the rule for phi is from nitrogen to alpha carbon. And the rule for psi is from alpha carbon to carbonyl. Okay, so those are the rules. You got to have them memorized, and then it's super easy. Let's start at the beginning and we just go from nitrogen to alpha carbon, well, this guy has to be 5. All right, let's find another nitrogen to alpha carbon, that guy has to be 5. Okay, let's find another nitrogen to alpha carbon, that guy has to be 5. Okay, from nitrogen to alpha carbon, that guy has to be 5. Nitrogen to alpha carbon, that guy has to be 5. Nitrogen to alpha carbon, that guy has to be 5. Okay. Easy? Yeah, once you know your rules, easy, right? All right, now let's label our psi angles. Psi angles, those are from alpha carbon to carbonyl. So we have alpha carbon to carbonyl. This is psi. Okay, so alpha carbon to carbonyl, this is psi. Alpha carbon to carbonyl, that guy's 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 psi. Easy? Yeah? Okay, cool. All right. So what else do we have to label here? Oh, peptide bonds. All right. Where is a peptide bond? That's where the amino acids are holding hands. All right. So you go from carbonyl to nitrogen. Yeah.
Why would it be side the last one?
Oh, alpha carbon to carbonyl and it's COO minus. Because if another amino acid were to come in, it would be a carbonyl. It like turns into a carbonyl. So like the oxygen leaves with the 2 hydrogens and a water droplet forms. That's how the bond forms. So it should be a side. All right, so let's see here. Sorry, what was I? Oh, peptide bonds. Okay, so where does the peptide bond form? Where you have the carbonyl with a nitrogen. So all we go is let's go find our first carbonyl to the next nitrogen. So this is our peptide bond. So this is where this amino acid is holding hands with this amino acid. So we'll label that peptide. All right, next carbonyl to nitrogen, that's another peptide bond. 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 Easy. All right, last thing we're gonna label is called, they want you to put a rectangle around the 6 atoms that are coplanar with a peptide bond. I'm not really sure how to explain this. I think the book only gave me like 1 paragraph, which I barely. So if you have questions about this, ask the professor. But I'm gonna go ahead and show you where it's at right now. Okay, we can do it right here. Okay, so you basically choose a peptide bond. Okay, so I'm going with this one. And if you notice here, we have 1, 2, 3, 4 atoms. They're asking for 6 atoms. So you just go from that alpha carbon to this alpha carbon. So here's my peptide bond. That's 4 atoms. We need 6 atoms that are coplanar with a peptide bond. So you just go from alpha carbon to alpha carbon. You basically just do this. So if they ask you on the test, oh, draw a rectangle around the 6 atoms that are coplanar with the peptide bond. Okay, and in terms of labeling, you're done. Oh, yeah, so they wanted you to draw the polypeptide KSF. So K is lysine. So you would just go to the alpha and you would draw lysine. Then you go to the next alpha and you draw the next R group and so on and so forth. So for example, lysine has 4 carbons and then an NH3. And then I just fill in the carbon, the hydrogen bonds after. So this is 2 bonds, it needs 4. because carbon makes 4 bonds. This carbon also has 2 bonds, but it needs 4, so you just add 2 hydrogens. This carbon, same thing. So you would just go in and fill in your R groups, and then you're done with this problem. You can do this on the test. They ask you. Yeah, hopefully. Okay. All right. All right, let me write down for myself, VGA to USB. That's what I have to purchase. All right, so now helical projection. We have an alpha helical projection. You'll learn more about this in lecture, but my students have had a really rough time understanding this, so I just wanted to explain a couple of things really quickly, super fast. So the way that it works is you remember an alpha helix looks like this. So you just basically fill in the atoms and it goes like, or the amino acids, and it goes like this. And that's just how it goes. So it's like looking at the helix from like top down. That's what you're doing here. You're just looking at it from top down. You fill in your amino acids, lysine, serine, and whatever else they give you. Then the rule for an alpha helix is n plus 1. Oh no, n plus 4 equals bond. So n plus 4 is bond. So you go to your first one and then you count 4. So 1, 2, 3, 4. So this first one and this fourth one are gonna bond. But they don't just want you to know that those two are bonding, they also want you to know how they bond. The carbonyl, because remember, this is just an amino acid. So it's got like the 4. There's the alpha carbon, NH3 plus, COO. So this is just an amino acid. They're all amino acids. So which portion of this amino acid will bond with this portion of that amino acid? So the way that it works is the carbonyl of the first one is going to bond with the amino group of the other guy. Okay, so I just wanted to show you that because a lot of students are really confused about that. What else do we have? Okay, let's move on to Ramachandran plots. So this I can just do on the screen. Okay, so Ramachandran plots are amazing. They are so cool because they're so easy. So first of all, the first thing you have to absolutely know how to do is draw a plot. You have to know how to draw it. So the first thing you do is you go ahead and draw your square, and then you divide that into 4s, okay? You divide that into 4s, and notice that here in the middle, we have a 0 angle. And then over here at the bottom, we also have a 0 angle. And your x-axis is phi. You see this? Your x-axis is phi. So sometimes, like on an exam, they can tell you, oh, your phi angle is 60, or your phi angle is -60, and your psi angle is 120. Well, you have to know which quadrant that falls in. And a lot of my students in the past didn't remember which axis was phi and which one was psi, so they would get it backwards, and then the answer would be wrong. So you have to know the axis. Just phi is the x, phi is x, and then you'll just remember that the other one is a y. So phi is the x-axis, and it goes from -180 to +180, and then the same thing for the y-axis. So once you know how to draw your axes and what goes where, all you have to do is learn 5 dots. So this is your key. Once you memorize these 5 dots, then you can answer any question and you'll get it right when it comes to Ramachandran plots. And this is why I love them. They're so easy. So basically, if you get anything right here in this dot region, this is a right-handed alpha helix. Okay, so it looks to be like minus 60 to minus 60. Because this is 0 right here. So let me try to draw the axes. Purple. Okay. So like we'll break this down into like 3. So this is like minus 60 maybe about. And then the other one is like minus 120 about. Yeah, you guys agree? Okay. So then if we look at the coordinates for this right-handed alpha helix, it looks like it's minus 60 and minus 60. Yeah. So on an exam, for example, they might ask you, oh, you have a phi angle of minus 60 and a psi angle of minus 60. What secondary structure are we looking at? And you're going to say, oh, that's easy. It's a right-handed alpha helix. Because remember, alpha helixes and beta sheets, they're secondary structures. So you have to know all your terminology. Okay. All right, so the next one, we have a left-handed alpha helix. So this dot over here, so that one down there was a right-handed alpha helix. Now we're looking at left-handed alpha helix. And that looks to be like at positive 60 and positive 60, okay? So you don't have to memorize the angles, but just know like which quadrant the dots are in. So here's a left-handed alpha helix, okay? So we know that the alphas are over here. And I like to draw them because when I'm first like memorizing things, oh my gosh. Second, okay. Please let me draw on there. Okay, so this is an alpha helix. Okay, this guy is an alpha helix, so I like to draw them. Okay. And then if we go to this last quadrant, guess what? Right up here in this top left-hand corner is where we have our beta sheets. Okay, so we have the anti-parallel beta sheets. So those are the guys that go like this, anti-parallel. And what do beta sheets look like? They just look like sheets, like that. And then we have the parallel beta sheets. So those guys, they all go in the same direction. And again, they're just beta sheets. So they just look like this, okay? And then that last dot right there is a collagen triple helix. They usually don't ask you about that. And then over here in this region, we have the right twisted beta sheets. So really it's just those 5 dots that you want to know. And I'm going to show you how easy it is to answer these questions. Once you have that there, it says, examine the 2 proteins labeled A and B. Which of the 2 Ramachandran plots labeled C and D is more likely to be derived from which protein and why? Well, what do we see here? Alpha helixes or beta sheets? Yeah, so we see a lot of beta sheets. And where do we know that those usually go on the graph? Top left-hand corner, right? So then we look at both of these graphs. Which graph has the most dots in the top left-hand corner? This one. Yeah, so that's how we know, oh, that's the right answer. Isn't that super easy? That's it. That's all there is to it. And then we look at the bottom one. So we see a bunch of alpha helixes, right? And where are those alpha helixes supposed to be? Either here or here. So we see a bunch right here, and we know that this is the alpha helix location. So that's got to be the right graph. Easy? Yeah. Don't you love Ramachandran plots? Even if they give you something a little harder and they tell you, oh, your phi angle is positive 60 and your psi angle is positive 60, and they give you like pictures, you're just going to choose the one that has a bunch of helixes, right? Or if they give you like graphs, you're going to choose the one that has all the dots in one of the alpha areas, right? So that's why I just memorized those 5 dots and your axes, and you've got this. You can answer any question with Ramachandran.
I thought that protein following like alpha helices, like the right-handed alpha helices, not left-handed. So why do dots show up in the left-handed alpha helices?
I don't think it's that, like, okay, anything in our bodies,
Yeah.
it's not like only, it's like mainly.
Oh,
Yeah. So there's a lot of things that are like rare, but in biochemistry 1, we only show you like the rules about like what we normally see the most. Because if we went into like all that, you'd have to learn like twice as much material. So that's what it is. But yeah, good observation. Yes, because when I also was thrown off at the fact that there was a few dots over there. Yeah, there's always going to be rare and exception. Okay, so great. You learned the backbone and you learned Ramachandran. Those are 2 of the biggest reasons my students would lose points on this material. So the next one. So this one, once you know the rules, you can do number 4. But I'll go through it super quickly because we need to get to 5 because that one's hard for students. Okay. So for number 4, they give you like a chain of amino acids and then they ask you, where might bends or beta turns occur and why? Draw the provided amino acid sequence in the bent form. Use the single letter abbreviation. Okay, so beta turns, they happen at proline. So you just go to the prolines because we know that proline makes the alpha helix unstable, but beta turns or beta structures, sheets, they love proline. They love it because it's what they turn. You know how like you have to turn, you have to make turns. Like right here is a turn, right here's a turn, right here's a turn. Like right here's a turn, right here's a turn, here's a turn. Guess who's at the turn? Like sometimes glycine is at the turn, but most of the time it's proline who's at the turn. Because he's so little and he's already like flipped kind of, and then he's stuck in that because the nitrogen is attached to, like this amino terminus is attached to the R group. So it kind of just like folds and then it doesn't have free rotation about this axis. And so it's like really sturdy and that's why it's perfect for these turns. Kind of get it, sort of. There's no rotation about this axis. Normally, the R group is free to mingle with other stuff, but in proline, it's not free because it's already attached and it's kind of like curved and it's very like, you can't move me, I'm stuck here. So that's why beta turns love proline at the turns. So for this question, all you got to do is you just got to find all the prolines. So the correct answer here would be 7 and 19. So if you need to draw the whole structure, I went ahead and drew it for you right here. It says draw at the turns. So that's this guy right here. Okay. Okay. Where might intra-chain disulfide cross linkages be formed? So intra-chain just means within the chain. So this whole thing is a chain. So where are you going to find disulfide cross linkages? Which amino acid is responsible for that? Cysteine, right? Because it has that SH. So cysteine is the one amino acid that is responsible for disulfide bridges. Okay, and that is a covalent bond that stabilizes 3-dimensional structures. If we didn't have these, then we would end up with 3-dimensional proteins that are kind of wobbly, that could easily be disrupted, and that could lead to disease. Because if you have a protein that does not have a beautiful, perfect 3-dimensional structure, it cannot perform its function. Okay, So cysteine residues are super important. Disulfide bridges, covalent bond, stability. Those terms that I just threw out there, you need to have them memorized, because if they give you a question where you need to answer in like sentences, you need to say all those terms. Okay? So good thing I'm recording, I hope. How can you tell if you're recording? Hold on, let me see. Stop share. Yeah, I'm recording. Okay. Cool. Okay, the last problem I want to cover is the Anfinsen experiment. Okay, if you guys want to take a picture of this, go ahead and do it now, because I'm going to erase the board. Once again, you're learning a lot in only 50 minutes. So good job. Okay, there should be enough. Anfinsen. So before Anfinsen, we as a scientific community were like, you know what? If you have a three-dimensional protein, but if you ever unfold it, you will not be able to fold it back again. It is not possible. And then comes Anfinsen. And what does he do? Well, he plays with urea, and he plays with beta mercaptoethanol. So again, our terminology, these are two of the terms we have to memorize. Urea disrupts hydrogen bonds. In other words, it denatures proteins. Beta mercaptoethanol, it breaks disulfide bonds. So if you remember correctly, last week we talked about two other reagents that do the same thing. Why didn't he just use those? Well, Anfinsen was from I don't know what year. He probably didn't have access to those reagents. So this is what he used and it worked. Okay. So here we go. Let's say that we have a three-dimensional protein and Anfinsen uses urea. What are you going to get over here? You are going to get it's going to be unfolded, right? It's going to be denatured, but any disulfide bridge that was there is still there. It's still attached because that bond is really strong. Those are covalent stabilizing bonds. So those guys are still there. But if he uses urea and beta mercaptoethanol, the urea is going to denature the protein. It's going to unfold it. And then the beta mercaptoethanol is going to go in and break those bonds. So your protein is going to be fully unfolded. And remember, it's going to be in a liquid solution. So it's going to be wobbling around all the time, the whole time, due to Brownian movement and just whatever, vibrations everywhere. Even if we try to be super stable in lab, when I'm working with stuff, I can't be. There's vibrations. Stuff is moving everywhere. So it's just imagine it wiggling around. That's important. But so far, easy? Yes, once we know our terminology, things are not so scary, right? This is easy. Okay, now the hard question. What happens if you add urea to your three-dimensional protein? Oh, sorry. Okay, I skipped something. Okay. So when you add urea, you have your three-dimensional protein, it unfolds it, and then this is where Anfinsen is really cool. He said, okay, I'm gonna remove the urea and see what happens. What do you think happened? What everyone said would not happen. The protein went back to its three-dimensional shape. And this blew away the science community because they were just like, oh my gosh, you add urea, it unfolds your protein, you remove the urea, and all of a sudden your protein is back in its perfect three-dimensional shape. That's phenomenal. So this told the science community that it is the sequence. The sequence of amino acids are what determine and hold all of the knowledge that you need to be able to make that perfect three-dimensional structure. Because remember, we're trying to cure diseases. A lot of these diseases are because the proteins did not fold correctly. So we need to know everything that we can about how proteins fold, unfold, and refold so that we can start examining in detail some of these diseases. So that's why this was huge. And this right here is what you need to get out of Anfinsen. Because of him, we now know that protein folding is dependent on the sequence of the amino acids, and that it is possible to denature and renature proteins. And these were his tools that he used, urea and beta-mercaptoethanol. Now, this is kind of like a puzzle here. So let's say you add urea, you denature, you add urea and beta-mercaptoethanol. Oh, hold on, okay. Urea and beta-mercaptoethanol fully denatured. So I'm going to say fully unfolded, fully unfolded. So then you remove both. But you remove urea first. Then beta-mercapto. So what's going to happen? Well, if you remove urea first, the protein is going to start folding back into its three-dimensional structure. Those disulfide bonds are still going to be broken, but the urea is, you remove the urea, so the protein wants to fold back into its three-dimensional structure. And then you remove the beta-mercaptoethanol. So now so we're going to say refold. And then we're going to say then the disulfide ridges reform. So because the protein was already refolded, those disulfide bridges are going to happen in the same exact places where they originally were. And that's what you want. However, now we have this condition where you have urea and beta-mercaptoethanol, so same condition, you have a perfectly unfolded protein, cool. Then now you're only going to remove, you're going to remove both. But you're going to switch the order. You're going to remove beta-mercaptoethanol first. first. And then you're gonna remove the urea. So what's gonna happen first? What's gonna happen first? Yeah, the disulfide bridges are gonna reform, and because this guy is just wiggling around in water, where are those disulfide bridges gonna reform? Whoever's closest. Is that gonna be correct? Probably not. And these are covalent, stabilizing, super strong bonds. So what's gonna happen to your protein? It is not gonna fold properly, and it is gonna lose its function, which leads to disease and possibly death. Yes. I'll read. So we reform the disulfide bridges in the wrong places. And then when you remove the urea, the protein refolds. Incorrectly. Okay, so you guys did a really good job today. You learned a lot. 3 very important, huge concepts in 50 minutes. So good job. I'm gonna go ahead and post this video maybe tonight, just in case you need to rewatch it